All right, so I would also like to thank the organizers for inviting me to speak about this today and on this fantastic panel. I'm really impressed with, I've learned a lot already. Hopefully I can add some more context to this as I speak about ethical challenges in the analysis of archived biospecimens and some of my sort of frontline experience in wrestling with some of the questions in this area, many of which come down to the adequacy of informed consent, which I think you've already heard us allude to. So just to remind us why we're talking about this, I probably don't need to do this for this group, but you take a theoretical case where we may have an exciting new biomarker that's determined to help us figure out taxane sensitivity in one disease like lung cancer, called it MWO for microtubules watch out. And we want to see if we can apply this data in breast cancer. So how do we do that? Well, you can launch a new study, and I'll get back to that, but maybe we have archived biospecimens with tumor blocks that we could turn to, or blood samples if it's a pharmacogenetic study that we could turn to. You might imagine that, okay, we've got this trial, we took over, tremendous amount of patient resources, over 2,500 women, randomized to one of two regimens, one of which is a taxane-based regimen, and we've got a secondary randomization to 4 versus 6 cycles of chemotherapy to address an additional question. What we learned from the trial is that paclitaxel is really not quite as good as AC, so it won't change the standard of care. 4 is about equivalent to 6, and that's fine. It was an important study, but those are the results. Now we have an opportunity to do something new with the data. We've got biospecimens from about 1,000 patients. We've got 10-year follow-up. We know that, for example, the disease-free survival was here if you don't stratify by this new molecular marker. So we want to ask the question, what would happen if we use this marker here? And let's just imagine that we can divide patients into MWO negative. We get these kind of results. And we can divide the patients that are MWO positive. And sorry about the little Mac conversion issue here. But we find that, wow, when you look at this biomarker, it selects a subgroup where the taxane was really important. They do much better than expected, and maybe 6 cycles even better than 4, and we'd, of course, advance the science. This is theoretical, but that's, of course, where we'd like to go with use of archived biospecimens. We'd like to change care. Wonderful. Now, what if the informed consent we obtained, however, was inadequate? What are we left to do with this scientific potential that's unaddressed? Either it doesn't describe the current purpose of the research that we're trying to address in the same or other diseases. It doesn't specifically mention the technology we're going to use. Maybe it sort of dances around that and talks about future research, but it doesn't directly say genetics, for example. It doesn't, maybe it mentions tumor genetics and says, well, we're going to do things related to the genes of your tumor, but it doesn't get into or even implies that we won't be looking at inherited or germline genetics. And what if it doesn't adequately describe the risks, either the privacy risk, the risk of discrimination on genetic basis that others have discussed so far today? So these are real world challenges that we face using our specimens. What would our options be? We could start out and organize a new 2,000 plus person trial. In this case, it would be difficult to do that. You'd have to give one group what's considered a non-standard and inferior therapy, wait 10 years for the results. We probably can't do that easily. We could try to connect new research biopsies from people treated outside of the trials and use our rapid learning systems to sort this out. We're not quite there yet in terms of those systems, so that might be a challenge. We could leave that, essentially, that kind of scientific knowledge on the table and say there's some questions that we're just not going to be able to answer. Standard of care has changed. And if there's other ideas, I wasn't able to think of them coming up with this. So we've got this challenge. Do we leave aside the scientific benefit, or do we somehow violate our ideal sense of informed consent? Is there a middle ground? So we know, people have been talking about this throughout the whole session, certainly today's session right here, that we're in this brave new world, which I think Angela's told me that I'm about 5 times too high here in my assessment of what it costs to do whole genome sequencing now. But we all know that it's changed tremendously from the billions of dollars to the few thousands of dollars. We're in an era of cheap, relatively, whole genome sequencing, an era of data sharing, and increasingly an era of international collaboration. I think that's another challenge that we need to address. We may have trouble coming up with standards that just apply in the US. We need to include our international colleagues as well. And of course, we know that there's groups working on this same challenge in those countries. We also have the concept of the idealized informed consent that's very important. It's the key to public trust in research and to having all of us continue to participate in biobanking and clinical trials. We want to explain the rationale, the societal benefits, potential direct benefits to our patients, the participants, the details of the study, the likely harms, and then the potential risks. And I think we have pretty good consensus that on all of these things are important. But there's a huge literature on how difficult this is, even in describing the trial we want to do today, where we know exactly the questions we want to address. We've got pretty well-defined harms. It's hard enough to achieve anything close to ideal informed consent. How do you do it for future unspecified research? And what do you do when you've got archived biospecimens with consent obtained under one standards, and now you're 10, 20, 30 years in the future? So the challenge is exactly that. Biospecimens from older generations of trials that simply did not use today's language in terms of informed consent to explain genetics, to explain the limitations of de-identification, no matter where we are in the spectrum of believing they're a real concern or not, to describe the data sharing and how that's done and what the limits of that are going to be. Is it going to be just a broader group of American investigators? Is it going to be international? What controls will there be on it? To explain the potential privacy risks that then come from that, and to describe uncertainty, which is an evolving area. We also know, and I think the other speakers alluded to this, that the biospecimens were ultimately, in all cases that I'm aware of, provided to help advance science and help future patients if they gave consent to future research. So I want to suggest that this issue of public trust that we worry about in terms of violating informed consent really cuts both ways. And I think just as we want to be careful that, boy, would this pass the smell test if the public learned about the data we're sharing here, what we're doing with these archived biospecimens? Well, what would the public think if they learned we didn't use this information to try to address the diseases that still plague us? So I'm going to talk through a couple of examples where I dealt with issues like this as part of the ethics committee of CLGB and now the Alliance. This is a multidisciplinary committee. There's members of the committee in the room here. There's patient advocates, clinicians, clinical researchers, folks with some expertise in bioethics, broad multidisciplinary group that doesn't actually have decision-making power, but is brought in to consult on difficult cases where the investigators or others raise some questions. So this is sort of course, the hypothetical trial I was showing you before. That example was fake, but the trial is real. CLGB 40101 was a trial of a little over 2,500 patients with early-stage breast cancer. They were randomized to the chemotherapy regimens I've shown here, secondary randomization, and it was optional for patients to participate in a GWAS study that was designed to look at genetic predictors of response or resistance to therapy. And the question came up that came to us on the ethics committee was, can we share this with NIH's dbGaP? We are in fact have some obligations potentially through research funding to share this data, but dbGaP doesn't want us to share it if it's not appropriate. So what do we do? And this was considered by the ethics committee. So what do? You go back and look at the informed consent documents. And what you find is that, of course, the study was initiated well before dbGaP was imagined. So the informed consent is not going to explicitly address this situation. It did say that participation was voluntary and people could opt out at any time. It did discuss future, what I'd call genetics and pharmacogenetics, so that was a known intentive part of the study in general terms, but it didn't specifically say genetics. It did discuss the idea of uncertainty regarding details of future research questions, so it did some things that we would all like. It also promised that there would be review within the investigative committees of CLGB of future research. So that's not exactly what dbGaP does. They've got a very rigorous review standard for who gets access to the data, but they don't normally process it all through CLGB or any other specific cooperative group. So it did not use, specifically use the words genetics or genetic information. It did not mention, sorry about the typos, did not mention sharing the genetic or the clinical data within a national database. It didn't mention privacy risks, and it didn't mention the potential for recontact that you might get in that database, still to be worked out. So it was problematic. So initially, we addressed the question. We said, okay, informed consent is not ideal. Can we reconsent patients? That clearly would be ideal. That's our ideal. If we've got any question about whether informed consent was adequate, go back and ask folks. And the appeal of this in this case was that it was an adjuvant trial. And it was an adjuvant breast cancer trial. So given the nature of this relatively low-risk population, you'd expect 80 to 90% of people to be alive and disease-free. You might be able to reach them. On the other hand, as we dug into this, and that was actually the ethics committee's initial recommendation after much deliberation, lots of consultation, was consent form is okay, could be better. why don't we try to recontact people and make sure that everybody who's going to participate in this is willing. But not only expensive, very logistically difficult. It's dubious about whether you even could do this with a large percentage of the group. There were concerns that in those who have experienced an adverse event, either death or recurrence, they might be upset. And that, of course, may lead some to differentially participate or not. There may be other factors that impact somebody's desire to participate. And ultimately, you would have a potentially cripplingly biased scientific sample. So what came back to us on the ethics committee was that really this is not, you can't find your gray zone way out of this and reconsent. It's going to be all or none. You either use the data from people with the limitations noted, or you just say, we can't do this. So we've published this, what our, a little more detail of what our deliberations were and the issues we thought about. But the key considerations we arrived at for making decisions in this area were to consider the potential societal benefit from sharing the data, to ask, was there at least informed consent for future research? That's sort of the low bar to get over, but if there's not that, you got a big problem. Was the uncertainty of risks from future unspecified research explained in any way in the consent form? Because we know that our appreciation of risks is changing. Did you at least make it clear to the subjects that we're not able to, at the time we're consenting them, explain all the things we might worry about or be aware of in the future? Was data sharing consistent with the goals and terms of the initial consent form? If the language wasn't perfect by modern standards, was it at least comparable so that somebody might have agreed? Is reconsent feasible? Of course, is a key issue. If it is, I don't see a strong argument against doing it. But ultimately, this question alluding to where does the public trust lie, will the expressed interests of the participants be better honored through data sharing or withholding the data, as really the ultimate question to address in each case. I'm going to talk about one more example that came up before our committee. This was CLGB 80101, so I can only presume it was twice as good as 40101, or that we have limited imaginations when we number our studies. Phase 3 intergroup trial of adjuvant chemoradiation after resection of gastric or GE junction adenocarcinoma. It was a little over 500 patients. There were two, I don't have the details, the regimens here. They were essentially equivalent. So the trial itself didn't find any difference. They were both variants on 5FU and radiation. But the important part is that we had samples from over 400 participants that were banked in the CLGB, later the Alliance Central Repository. And the question that again came before us, similar to the other one, was, hey, can we share this with the Cancer Genome Atlas project? So many of you may know this is a 5-year intensive effort to molecularly characterize cancers and compare the molecular profiles with the clinical data to try to really jump the field ahead. And the feeling of one of the investigators very familiar with this area of research was that given that there was no preoperative therapy here, so these were all samples taken just from the disease before they went into the trial, but with the excellent clinical annotation that you get from participation in a large cooperative group trial, that the tissue specimens from this trial represented a unique and really unprecedented resource. So the scientific stakes here were high for being able to use this. But that said, was it ethical? So the TCGA gave, I thought, some very helpful guidance in this area. They said, if you're going to provide data to us, the informed consent document has to do a couple things. It has to describe genetic or genomic research. It has to explain the concept of data sharing and in broad terms and make it clear that there's going to be use by individuals other than the PI and the people that the patient or participant was already working with. It has to explain the possibility of future research and ideally explain, because of how they plan to use the data, research beyond cancer research, and explain that research could theoretically result in commercial products, because of course that's one thing that the Genome Atlas project may lead to. And it had to explain the risk of loss of privacy and confidentiality and explain any mechanisms that were going to be taken to protect that. So a pretty high bar for what it had to contain. So we looked very carefully at the informed consent documents, the language. I think if you look, this is not unique to this trial. If you look at any informed consent document I've sat on, cancer protocol review committees and IRBs, whether it's institutional or multi-institutional, you see similar language. We can clearly do a lot better. But it said things like, we would like to study cells from your tumor for research only. We'll not study inherited genes in one place. And do you opt in or out? These were all opt in or out questions. I've just checked them all off here, but either I agree or disagree that my tumor can be used for this. The next question and the next statement was, We'd also like to study whether substances in your blood, sometimes called tumor markers, are related to the way your body responds or doesn't respond to chemotherapy you receive. Tumor markers are inherited through your family, could be passed on to your children. These are also called genetic studies, so I can tell you with my current understanding, this is very confusing to me. We're talking about inherited genes and tumor markers. I don't think of these as the same thing. So I can imagine it's probably confusing to lay public. But again, you either consent to this or not. It's a little contradictory to what was mentioned up above. And then a third one was I either agree or disagree that my DNA may be used for future use in future DNA research studies, with, of course, some more explanation about what DNA was. My tissue or blood may be kept for future unknown use in research to learn about, prevent, or cure cancer. And a final opt-in, opt-out. They could check any one of these in any combination they wanted. It can be kept for research about other health issues like diabetes, Alzheimer's, heart disease. So that's what we had. And in terms of reconsent and things like that, we quickly learned the study was open at over 500 sites. There was no central IRB, so it was really not possible in any way to go back and approach these individual sites, put it through all the IRBs, and try to find these patients again. It's also a disease where I think Rich can correct me if I'm wrong, I think the 5 or 10 year survival was like 40% maybe, something along those lines. So you wouldn't be able to reach a lot of these people even if you wanted to. So after considering all this, although again we left some science on the table, we said that most consistent, we wanted to take advantage of this resource. We also didn't want to violate any principles of informed consent, but that clearly for people who had answered yes to all of those, they were intending to give their biospecimens and say, use them broadly, use them for other diseases, et cetera. And fortunately, not everybody, but the majority, the vast majority, 312, so about 75% or more, had given consent to all 5, and that's what we decided would be used in this case. Some could say you should have set the bar higher, some could say lower, but that's where we were, and that's why I present this example. So in closing, really, what can we say about the future? The only thing I can say with any confidence is that we're not going to be able to predict the questions we're going to want to ask in the future or what the technologies will be. I'm pretty sure that, like in the movie Sleeper, we'll have, robots in the home and jetpacks, but beyond that, I'm not at all sure what we're going to have access to. And so I think we've got a bunch of issues to address, many of which have been mentioned already by my colleagues here. We clearly need to, when we're dealing with uncertainty and imperfect informed consent, come up with a transparent and multidisciplinary process that our participants would be happy with to review the use of their archived biospecimens and decide what we can or can't do with them. And I think we clearly need research in this area, so it's great. There isn't a lot in the literature right now. It sounds like what the eMERGE group is going to do, surveying 16,000 patients on these exact questions, is going to go a long way towards informing us how the public really thinks about this. I think also we really need to plan for abuse. I mean, look what's going on with the NSA, and people are going to be able to hack into this data. I think if we assume that we can describe the risks and protect people and put in layers of protection, we're setting people up for false understanding. So I would like to see us put in the laws and regulations to address that, assuming there will be problems. Things like laws against discrimination in health and life insurance, government employment, other government policies and workplace employment, and then criminal penalties, just like we discovered as we got into cyberspace, that the law had trouble catching up with the technology. Let's try to get ahead of it in the law here, prohibiting the things that we don't want done. And then we obviously, as others have mentioned, need to try to continually prospectively improve informed consent for future unspecified research. And I think I'll leave it at that. We'll have time for questions. Thank you.